All right, our next speaker is Jesse Engreitz, who's an assistant professor of genetics at Stanford University and also a member of the NNFC Center for Genomic Medicine here. Jesse.
Thanks so much. Really happy to be back here at the Broad and see so many friends and familiar faces. I'd like to share today some of our two recent stories trying to map the regulatory wiring of the genome to link variants to functions. So building on this beautiful framework that Melina laid out now 10 years ago, we know that each of these variants that we've discovered regulate some gene in some cell type to affect some pathway to affect disease. And if we could map that variant to function connection, we might discover clues that help us develop new drugs. There are, of course, beautiful examples from this community that have illustrated this paradigm. One that I love is VJ and Stu Orkin and Dan Bauer's work to characterize variants associated with sickle cell or fetal hemoglobin, where they showed, of course, that there are non-coding variants in an erythroid-specific enhancer of BCL11A, which VJ showed is a repressor of fetal hemoglobin. And that if you target CRISPR to cut at this enhancer, you can create a therapy for sickle cell disease. And I wanted to share this example to illustrate three key points, one of which is that most of these disease variants are non-coding. These disease variants tune gene expression in specific cell types, and this is proof of concept that editing regulatory DNA has therapeutic potential. So we're working on a number of different approaches to systematically accelerate this process and to more rapidly convert variants to functions, to medicines. And I wanted to share two particular stories, the first of which is to think about how could we interpret non-coding variants by identifying all of the enhancers in the genome and linking them to their target genes and cell types. So, of course, there are a few different approaches to link non-coding variants to their cell types and target genes. One might be to apply single-cell EQTL approaches, like we heard about today. The challenge here is power and scale. We could also use CRISPR to go in and edit variants or perturb enhancers that contain variants. And of course, here there's a similar challenge, is that these non-coding sequences can have very cell type specific effects, and we can't possibly CRISPR every possible cell type in the human body. So what we need instead, or in addition to these two approaches, are really good predictive models that would allow us to take a particular non-coding variant and look up its function across nearly any cell type in the body and identify the cell types in which that variant might act, as well as the target genes. So one simple approach that I developed together with Charlie Fulco when we were both here at the Broad is called ABC, or the activity by contact model. And the idea here is that if you simply estimate enhancer activity using bulk DNase or K27 cell ChIP-seq data, and you multiply that by the 3D contact frequency between an enhancer and its target promoter, which you can measure with Hi-C, you can actually predict many enhancer gene links and begin to build these genome-wide maps across many different cell types. The challenge with this model is that it's not 100% accurate, and it relies on these bulk epigenomic measurements that are difficult to acquire in the many thousands of cell types and states in the human body. So for this project, we teamed up with Robin Anderson's lab, and in a project led by Maya Sheth in my group and Weilin Chu here, we thought about, could we use single-cell data to predict these enhancer gene links instead? Could we leverage the growing amount of single-cell epigenomic data to build predictive models to learn enhancer gene links in any cell type? So the first step here was to build a large gold standard data set. Good first step when developing a predictive model. Here we're using CRISPR-I to perturb many different enhancers and measuring their effects on target genes. And for this study, we used an integration of many previous studies that was led by Andreas Geschwin in my lab, where we integrated some of the data we had collected with data from Jay Shinder's lab and Lars Steinmetz's lab, reprocessed and harmonized everything to create a data set of about 10,000 gold standard examples where we've landed CRISPR-I in a particular element and very precisely measured the effects on gene expression. And The effect sizes of these elements span a large range, from 0 to almost complete repression, and then the red dots are the regulatory enhancer gene connections, and the gray are the ones where you perturb with CRISPR. We had good statistical power, but you don't see an effect. So we can use this data set to evaluate different predictive models. And the first thing that we did was compare these CRISPR data to various models that have been previously developed to predict enhancer gene regulation from single-cell data. And so here's a precision recall curve predicting the CRISPR data. The perfect model would be up here. If you simply link every element to the closest gene, you get it right about half of the time. If you link elements to genes based solely on distance, you get this curve here. If you build sort of a poor version of the ABC model where you estimate activity using a tag-seq only, and you estimate contact with a function of distance, you can do slightly better than distance alone, but not particularly well. And it turns out it seems like a tag-seq in particular is not a great measurement of enhancer activity compared to DNA-seq or to K27 cell chip-seq. And then in gray are a number of other models that are based either on correlation between accessibility and gene expression across single cells or cell types, or based on regression models that are trying to use accessibility at multiple nearby peaks to predict expression. And none of those models did particularly well at predicting the CRISPR data. So the model that we developed is called single-cell E to G, and it works like this. So we're going to take a particular single-cell data set that has many different cell types. We're going to look at one cluster or group of cells at a time, and within that one group of cells, we're going to compute a number of different features. First, we're actually going to compute the ABC score from the pseudobulk attack in here, and then Wayland developed an approach to integrate information about accessibility, gene expression correlations with ABC that actually strengthens the predictions of this score. We also add in a few features like distance, the class of the promoter, which we've shown tunes responsiveness, and other features that are sort of based on molecular mechanistic studies of how enhancers work that we thought would be important. And then we train a very simple logistic regression classifier directly on the CRISPR data in cross-validation. So this is an 8-feature model. It's not a billion-parameter foundation model, and fortunately, there's no chance of it gaining sentience soon. So here's the performance of this model. It actually does quite a bit better than these previous approaches in cross-validation in the CRISPR data set, showing that when we combine this ABC score with other features, we can indeed boost the performance and now accurately predict the CRISPR data from a single-cell multi-omic experiment. One of the limitations of this analysis and of many of our previous work is that a lot of this CRISPR data is in a single cell type in K562s. And so we were also wondering whether this would generalize to other cell types. In theory, the model which we've trained on K562 cells should now be able to take in multi-omic data from another cell type, transfer to that cell type, and make cell type specific predictions about which enhancers regulate which genes. And so to test this, we collected and again aggregated a number of different studies that we and others have done since we trained the model, and created a held out data set that's about 4,000 examples across 5 different cell types. And when we evaluate single-cell E to G in this data set, it also does quite well, much better than distance or the ABC model, and much better than these other simple correlation-based approaches. So we're now really excited that we have a model that we can apply to many different cell types and build genome-wide maps of enhancers. And just to illustrate what we've been finding when we are starting to use these maps to interpret variants, we've been interested since moving to Stanford in understanding the genetics of heart development and heart diseases. And in one study in collaboration with Will Goodyear's lab, we collected a 800,000 cell single cell atlas of human fetal heart development between 6 and 22 weeks of age across 41 different samples. There are 90 different cell types and states in this atlas, including all of the major cell types in the developing heart. as well as many subtypes that we're starting to recognize now that we have sufficient depth and really deep attack sequencing in this single-cell atlas. So for example, among the endocardial cells, if you subcluster the endocardial cells, you find clusters of cells that are specific to particular spatial locations in the heart. So the endocardial cells along the lining of the atrial septum look very different than the cells that are lining the inflow side of the valve, and those look different than the outflow side of the valve. And so they're very specific, and in this case, spatially distinct gene regulatory programs at play in these different cell types. So here's an example where we are using these single-cell enhancer predictions to interpret a variant that's associated with atrial septal defects, as well as with various quantitative measurements of valve structure. And when we look up this variant across 90 different cell types in the developing heart, here are just a selection of different cell types. We can see that this variant is in an intron of a gene called GSR2. It overlaps in an enhancer that's predicted to regulate both GSR2 as well as this other gene, Wnt9b. And specifically in endocardial cells in various locations in the heart, and most strongly in endocardial cells on the inflow side of the valve, as well as in the atrial septum. Just as one sort of confirmation that this is working, we cloned this sequence in a mouse reporter assay together with Len Pinocchio's lab, and it's a little hard to see here, but basically this human enhancer sequence lights up in the outflow tract here, and when you section this, you see that indeed this enhancer is active in the endocardial cells, which are the single cell lining along here, the developing valve. So this is a great example, I think, of a variant that we've puzzled over for a long time, and that we could only interpret once we had really cell type resolved predictions of the regulatory genome. In this case, we have no good cellular models of endocardial cells, and certainly not of the different subtypes that exist in these different spatial locations in the heart. And so it's only by looking at the cell types in their native tissue environment in vivo with these predictive models that we're able to interpret and start to understand what this variant is doing. So we would love for you to try this out. This model is generally applicable to any single-cell multi-omic data set. We also have a version that works with single-cell attack only. And of course, as we expand these data sets into many individuals in health and disease, these types of predictive models, as well as the one that Jesper described earlier, will be able to make inferences about what variants are doing across many different cell types and contexts. We've also recently just made public an E2G browser. So if you go to e2g.stanford.edu, this extends the open target interface. You can type in your favorite variant and look up its predicted effects across about 1,600 different predictions that we've made across all of ENCODE and many single cell data sets. So let us know how that works for you, and we're curious to see what we can learn. So in the second half of my talk, I'd like to share some of our work starting to explore if we could rewrite regulatory DNA to reprogram gene expression in desired ways. And this is work by a graduate student in my lab named Mike Montgomery, who just defended, who was thinking about Casgevy and this example of BCL11A, where, of course, that involves cutting at a particular TF binding motif. And in theory, you could also imagine adding a new TF binding motif, and would that downregulate or upregulate gene expression in a specific way? Of course, the genome knows how to turn genes up or down, and if we wanted to recreate that for various therapeutic approaches, it would require us to have a really good understanding of where to edit, which types of edits to introduce, as well as a better understanding of how programmable this all is. So we thought it was a good time to start thinking about this when Mike joined the lab because of a few developments. One, of course, which was the introduction of prime editing, which for the first time made it really efficient to delete or insert, let's say, 10 nucleotides at a time, which is about the size of a transcription factor binding motif. There were also new sequence-based models for learning the language of the genome and how DNA encodes epigenetic states, such as Informer, that made us think that perhaps we had a better chance of figuring out what we should write into the genome. And what was missing was a high-throughput screening approach that would allow us to measure the effects of all of these edits, ways to adapt these models to design edits, as well as a better understanding of how programmable the gene regulatory sequences actually are. So Mike teamed up with a postdoc in the lab, Gabby Martin, to develop a method that we call variant effects. And the idea is that what variant effects enables is to introduce hundreds of edits into a particular enhancer or promoter sequence, and quantitatively and precisely measure the effect of each of those edits on gene expression, even in cases where editing is inefficient. So the way this works is that we'll design a library of pegRNAs that will recruit the prime editor to a particular enhancer or promoter, introduce many different edits, create a population of cells, including heterozygous and homozygous edits of each of these different sequences. Then we're going to use RNA fish to stain cells based on their expression of this nearby gene of interest, sort them into bins, and then sequence the cells in each bin to read out the distribution of edits in each of those expression levels. So for example, if you have an edit that decreases expression, you'll see that that edit has a much higher frequency in the low expression bin and a lower frequency in the high expression bin. And so this enables looking at hundreds of edits in one element. We're looking at hundreds of thousands of single cells carrying each edit, sort of allowing us to average over a lot of the clonal variation that can occur. This accounts for inefficient editing because we're sequencing the edited site and directly reading out the edits that are created. And there's other tricks that we use to get precise effect size estimates, which is really what distinguishes this method versus many of the others that have been developed. So, to illustrate that this works, we took a particular gene, PPAF, which we were interested in for its role in inflammatory diseases, and just as some positive controls, we designed small 4-base edits right at the first splice donor in PPAF, which we thought should break splicing and lead to non-sense-mediated decay. And so first, we did this the old-fashioned way. We introduced each of these pegRNAs one at a time into cells, picked clones, isolated and grew up those hundreds of clones, and measured their effects by qPCR. And indeed, each of these edits decreases expression of PPAF by about 80% compared to many wild-type clones. Although you can see that each of these dots is a clone, there's quite a bit of variability, which is just sort of how the amount of variation that's introduced when creating clones of this particular cell line. So now we can do the same experiment with variant effects, where we're going to introduce these 3 edits as a pool. And so here, for example, each of these edits is present at between 10 and 15% frequencies with 40% total editing in the pool of cells. We sort them using RNA flowfish against PPAF, sequence by PCR this edited site, and then look at the frequency distributions of each of these different edits across the different expression bins. And so, for example, compared to the reference, each of these edits is much more frequent in the low expression bin versus the high expression bin. We can plug these into sort of a maximum likelihood estimator to figure out what the original effect sizes would be, and learn that according to this estimation, each of those variants does indeed decrease expression by about 80%, both in the sort of pooled format as well as when we do this individually. And here, each dot is one of the biological replicates of this experiment, so basically one pool of cells that's been independently edited. And so these are much tighter because in each of these pools, we're looking at many hundreds of thousands of clones carrying the same edit. So we repeated the same type of analysis across edits of many different effect sizes. And here's sort of summing up a lot of TC, a lot of like tens of thousands of clones and many qPCRs, to show that across many different edits, the effect size that we measure in clonal cell lines with homozygous edits matches the effect size that we estimate with variant effects in this sort of pooled screen that includes cells of many different genotypes. So this was very satisfying, and hopefully that means that in the future we don't have to individually edit quite as many of these, and that we can trust the effect sizes that come out of this experiment. If the experiment says that this edit leads to a plus 100% effect on expression or doubles expression, then we can in fact believe that measurement. I'll skip over some of the, I'll just really briefly, the math for this is involved, and just for future reference, a few of the key pieces are that we have to use a precise editor. Here we're using PE2 rather than PE3, because if you start throwing in unexpected edits, that creates correlations between edits due to the, a single editor creating two different alleles in the same cell. So in order for this to work, you do have to use a precise editor that only creates the single desired edit. Okay, so now that we have this variant effects tool, we decided that to take it for a spin and see if we could start to reprogram gene expression in a specific way. So we tried a couple of different strategies, and the first strategy was, let's look at the promoter of the gene. And we had previously done some experiments to find a few really efficient PEG RNAs that worked with up to 80%, 90% efficiency. And so we took one of those PEG RNAs and we decided we would just insert a bunch of different transcription factor binding sites into this particular site in the promoter. Here we chose these 41 different sequences to match the consensus sequences for transcription factors that we thought were active in this particular cell line. So we cloned these into the PPAF promoter, do the pooled prime editing, measure each of their effects. For example, maybe we're inserting a motif for MYC or for FOS. And here's the results of this pooled experiment. So the effect size on PPF gene expression across two biological replicates of the experiment, the replicates are really highly concordant, and many of the variants do in fact manipulate gene expression, but not by particularly impressive amounts. So for example, inserting a MYC-MAX motif decreases expression of PPAF by about 10%, but maybe not exactly what we were looking for in terms of thinking about potential therapeutic applications. So we explored a second strategy, which was to leverage computational models to design stronger edits than we could trivially think of ourselves. And in this particular analysis, I'm going to show you work that was done using the Informer model developed by Ziga Avsek and David Kelly at Calico. And this was work by Johannes Linder, who at the time was a postdoc in Anschel's lab and then moved on to Calico himself. And the idea behind this particular model is that it is using a few hundred kilobases of DNA sequence to predict different measurements around the promoter. In this case, we're going to look at the predictions of PPAF cage or gene expression in this particular cell type. And what Johannes did together with Mike is to use the Informer model to create what's called a simulated annealing design process, where we would take the wild type sequence of the promoter, we would probabilistically choose to either insert a base, delete a base, or swap a base, create a new edited sequence, score it with the Informer model, and then accept edits with the desired effect with some particular probability. And the particular novelty of this approach is that we're using this iterative process, but also constraining it to only edits that should be efficiently delivered with prime editing. So instead of redesigning the entire promoter, we're only going to tweak a few bases here and there and see what we can do with very small edits that should be very efficient with prime editing. Okay, so in this experiment, we targeted 5 different high-efficiency sites in the PPF promoter, ran the design procedure, tested them with variant effects, and again, 2 biological replicates, the effect sizes are really highly correlated, and we did actually get a small number of edits that lead to up to a 2-fold increase in gene expression, or as low as I guess this would be a 7-fold decrease. So for example, if you insert a ZEB1 motif, which is a known repressor, you can reduce expression quite dramatically at this really highly expressed gene. And if you insert a composite ELK1 MIB binding site, you can increase expression. And this is also a good example of the model doing something that we didn't trivially think of ourselves. In this case, it's inserting about 10 base pairs, and it's creating this composite motif using a few of the sort of endogenously encoded nucleotides in the genome on each side. So it's actually creating a composite element that wouldn't fit in the 10 base pair constraint that we gave it. So we can, in fact, turn expression up or down by very large amounts with very small edits. There's another preprint from Keith Jung's lab that observes something similar. And so we think that this could be really useful in the future for both understanding gene regulation, the principles, how you turn genes up and down, but also potentially for gene therapies for certain types of diseases. One type of disease that we're thinking about are haploinsufficiencies, where you inherit one loss-of-function variant from one parent and you have one good copy of the gene, meaning that you have 50% the normal dosage. And because these are loss-of-function variants, different patients with the same disease will all have different variants. And so if you wanted to trivially correct the mutation, you'd have to have individual editors for each person. But instead, maybe you could instead imagine a therapy that upregulates the good copy of the gene by twofold, restoring the proper dosage, and the same gene therapy reagent could be useful for many different patients with the same disease. So there's a long road toward realizing that. One of the challenges is that while we were ultimately successful in finding edits that upregulated gene expression, the predictive models still aren't amazing. So here's a comparison of the measured effect on gene expression from variant effects with the predicted effect from Informer. And you can see that the single edit that upregulated gene expression quite a bit, actually Informer doesn't think it should have done anything. So there's a lot more to do here to figure out how to improve the models, how to improve the design procedure. Will it work differently to edit promoters versus enhancers? And we're interested in digging into this further. But for now, we have an initial platform combining this variant effects experimental method with sequence design that we think will be really useful for exploring this. So to summarize, we're really excited about understanding the regulatory genome to accelerate this process of connecting variants to functions to therapeutics. Would like to thank everyone in my lab as well as our collaborators for their support, including seed funding from the Variant to Function initiative here at the Broad over 8 years ago. Thanks very much, and I'll take any questions.
Terrific talk. Question. I'm going to ask a question, then they'll get organized there. But you snuck something in there that was, early on, you said that attack is not so good in the E2G. DNase is much better, and I think you've told me that K27 acetyl is maybe better, which is disturbing because a lot of us are using deep learning models to predict attack, and we were hoping then to be able to predict E2G from that. What are we missing in the attack? What's the problem?
Yeah, I don't know for sure, but what we suspect, so the specific statement is that if you simply count attack reads in a peak versus counting DNA-seq reads in a peak, DNA-seq seems to be a better estimate of activity when you plug it into these models. And I think that's because when you have really dense TF binding in the most active enhancers, the TN5 enzyme is just bigger than DNA-seq, and so it can't get in and make as many cuts. And so it ends up missing out on the really high activity elements. But hopefully that information is still there once we add in sequence and use sequence models to start interpreting which transcription factors are binding, and we're going to have to learn how to infer which transcription factors confer enhancer activity versus accessibility.
That was a really good talk, thank you. I was confused about this last edit, where the Informer model thought it shouldn't do anything, but it did, it doubled activity. How did it end up in your test set, actually, if Informer didn't think it would do anything? But you used Informer to pick the test set, right?
Yeah, so I didn't explain this detail, but we designed edits to activate the gene in 2 different cell types. So in this case, it thought that this variant should activate in the other cell type, but it actually did more strongly in this one.
Okay, thanks.
So I have a question about gene edits, let's say for some variants, you tested to try to upregulate the gene expression, you see maybe less than twofold or effects is weaker. Do you think it's due to, let's say, for the wild-type cells, two alleles? Did you test some edits using, let's say, heterologous, with the gene was downregulated first and then try to upregulate it to restore the gene expression? Will the effects will be larger compared to the wild-type context?
Yeah, that's a good question. I guess it comes down to whether, there's non-linearity in the promoter that controls how it responds to different levels of gene expression. As far as we can tell from other studies where we've synthetically combined enhancers and promoters in many combinations, is that most promoters don't seem to saturate and that you can always turn them up farther with enhancers. But certain promoters are just intrinsically less responsive. So there must be some point where like the polymerases are firing so quickly that you can't turn up a gene farther, but it doesn't seem like we've hit that point for most human promoters.
Hello, Jesse. I'm wondering in the SC-E2G model, how do you incorporate the bulk high-C data in the single-cell attack-seq and RNA-seq data? Because the cells are heterogeneous sample. If you measure high-C, there are a mixture of many structures, but how to use that to predict the specific cell type, specific enhancer and promoter interactions?
Yeah, so to estimate contact in ABC, there are a few different ways. The best is if you have really deep, let's say micro-C or intact high-C in your exact cell type. That leads to a boost in performance. If you don't have that, then what we do is we use an average high-C map across 40 different human tissues, which will capture basically the average relationship between distance and contact, as well as, let's say, conserved CTCF boundary elements that create specific loops at longer distances. So in all of these predictions, we're using a cell-typed average high-C map which is the same in combination with cell-type specific attack-seq data.
That's great. Thank you, Jesse.
Thanks a lot.